Assalamu alaikum friends. Now we will discuss entirely, and we also discuss the fluoroquinolones and cephalosporin generations. But before that, we should have a broad overview of all the generations and their categories. So in antibiotic classes, we will discuss class, gram, means the antibiotic which we are using is effective against which type of bacteria, either it is gram positive or gram negative, their examples and mechanism of action. So in minoglycosides, we see there are different types of drugs we named in the previous lecture, which are gentamycin, neomycin, streptomycin. So these drugs are effective against gram-negative bacteria, and the mechanism of action is to inhibit the protein synthesis, protein formations in the bacterial cell. So it kills the bacterial cell by inhibiting their protein synthesis. So the other drug is in this category, in this class, is cephalosporin. Cephalosporin is effective against both gram-negative and gram-positive. Its example are cefazolin and cefadroxil. These two drugs, in the next lecture we will discuss about the cephalosporin generation as well. The mechanism of action is to inhibit the cell wall synthesis. As we know that human cells do not contain cell wall. So this drug, cephalosporin or the penicillin, these both drugs are very selective and safest drugs that are used for humans, because human cells have no cell wall, so they selectively kill the bacteria that have cell wall. Either it is prokaryotes, eukaryotes, all bacteria contain cell wall. Now we jump to the penicillin, which is effective against gram-positive and gram-negative bacteria. Their examples are penicillin and ampicillin. Penicillin V, which is a natural penicillin, ampicillin, which are synthetic penicillins, which are made in the by just changing their derivatives. So we discuss about the ampicillin and amoxicillin, which are some drugs are methicillin resistant penicillin, and some drugs are very effective against methicillin, streptococcus aureus bacteria. Their function is same, their mechanism of action is same as we discussed in the cephalosporin, to inhibit the cell wall synthesis. That's why these two drugs, these two classes of drugs are very safest and selective for use. Now we will discuss about tetracycline, which is effective against gram-negative bacteria. Its examples are doxycycline and also tetracycline. These drugs are mostly given in a certain condition, like in ulcer. We will give as combinations like tetracycline, amoxicillin, and some proton pump inhibitors. The mechanism of action are to inhibit protein synthesis. We know that there are two ends of the proteins, which are 30S and 50S. So it will inhibit the protein synthesis of 30S binding site. The other drug is quinolones, or we can say fluoroquinolones. Both have the same name for the same class. It is effective against gram-positive and gram-negative. In short, we can also say that those drugs that are effective against gram-positive as well as gram-negative are broad spectrum. But we cannot clearly or confirmedly we cannot say that it really it is broad spectrum or not, because in gram-positive and gram-negative it can only cover 1 to 2 bacteria. If it covers a long range of bacteria, gram-positive and gram-negative, then we can say that it is a broad spectrum antibiotic. So in this drug class, quinolones and fluoroquinolones, we have example ciprofloxacin. They inhibit the DNA replication, topoisomerase 2 and 4. Topoisomerase is especially an enzyme that unbinding the DNA or remove the coiling, supercoiling of the DNA. So by inhibiting this topoisomerase 2 and 4, they can easily kill bacteria by inhibiting the DNA replication, because the DNA will not is a double helical structure. If it unwind, then bacteria can easily replicate itself. But if these drugs inhibit the DNA replication by inhibiting the topoisomerase, which is an enzyme that helps the DNA to unwind, if that enzyme is blocked, it definitely cannot replicate itself and bacteria will die. Macrolide is effective against gram-positive bacteria. Erythromycin is an example of this. Inhibit protein synthesis. 50S site. Sulfonamides against gram-positive and gram-negative bacteria. Sulfamethoxazole is an example. Inhibit foliate synthesis, that is why they are called anti-foliate drugs. They inhibit the formation of folic acid. Glycopeptides, gram-positive bacteria, vancomycin, inhibit cell wall synthesis. As we discussed in the previous topic, vancomycin is a class of others. These drugs do not come in any of these drug classes. These are the other drug classes, not to be confused with the aminoglycosides, because they have mycin name in them. In these two drugs, like cephalosporins and penicillin, they have the same ring, which is a beta-lactam ring. Beta-lactam ring in them. So, due to their structural similarity, they also have some similarity in their mechanism of action, like inhibiting the cell wall synthesis. So, we will discuss all the antibiotic classes. Now, we're going to discuss about cephalosporin and fluoroquinolones